Okay, welcome everyone to our meeting, Resist Believe Toronto chapter. Tonight we have as our guest speaker Rick Piercy, who's probably, the weather for him today is probably cooler than for us. He's in California. I don't think he's got it as hot as we do today.
Rick, that would be unusual for Southern California.
Rick is a semi-retired biology teacher. He's taught in high school and community college. And he's recently took some courses at Reasons to Believe, and Hilmer was in his class, and that's how we found out about him. So we asked him to share some of his insights. He's been associated with Reasons to Believe for 15 years. And the course that he took with Hilmer was Biochemical Design, taught by Buzz Rana. And so he's also been doing several classes in his local church and having dialogue with in his church. Some of them are young earth and some are old earth. So I'd like to welcome you, Rick, and I'm going to pass control over to you. Let me see where this is here. How do I do this here? Make you the co-host. Okay, you're going to be the co-host.
Okay.
And if others can put all their mics on mute so that in case there's any background noise, it won't affect Rick. Rick, just go ahead and start when you're ready.
Okay. Should I just begin sharing my screen? I'm not sure how to do this.
Whatever you want to do.
Okay. All right, does everybody see my PowerPoint slides? Are we good? And can everybody?
It's visible.
And can everybody hear me fine?
Oh yeah.
Okay, great.
Yep.
Well, welcome. I really appreciate the opportunity to be here with you guys. From Southern California to Toronto. This is always a interesting way to communicate. I'd like to just go ahead and open up in a word of prayer before we begin. Our Heavenly Father, I just want to thank you for this opportunity for all of us to come together and to just learn a little bit about what you designed and created at the cellular and biochemical level. I thank you for all my brothers and sisters in Toronto, Lord, and our desire is to share our faith with others, Lord, the hope that we have in you and your Son, Jesus Christ. So be with us this time, Lord. Open up our minds to what you have created. Show us new ways to share our faith. Open up new avenues for us to do that. Bring us into contact with those people that maybe have doubts about you, who are seeking you, and skeptics and others who have a different worldview. So we pray and ask this all in your Son, Jesus Christ's name. Amen. My title for today, what I want to talk about, is called Biomolecular Machines, a Contemporary Watchmaker Argument for the Biblical God as the Master Designer Bioengineer. And first of all, a little bit about my background. I do have a master's degree in biology. Interesting, my training is in ecology, not biochemistry, although I have a chemistry minor. And what led me to this kind of presentation and this topic is the class I recently finished on biochemical design, and it was just totally amazing to me. You know, like most sciences over the past 20 years, but especially molecular biology has just gone off the charts as far as what we've learned about the complexity of biomolecules and cells. And so what I selected were two what are considered cellular machines. In fact, they're even called machines by molecular biologists. I'm sure most of you have heard of them. One is the ribosome, which is the protein factory of the cell, and another is an enzyme involved in energy production. It's the chief power manufacturing unit in the cell, and it's called ATP synthase. So that's what I'm going to focus on, but I'm going to begin. I have a couple, several embedded videos, so hopefully we'll get them to work. The format is I'm going to go through some material for about the first, well, first of all, it's kind of divided in two parts, the ribosome and ATP synthase. And I'm going to, as I said, I have a couple of videos embedded in that, but I'll also stop for questions. And again, I'm not a biochemist. I'm sure some of you may have probably a better background than I do, but what I want to focus really are on the apologetics of how we might use this as believers. And also, I want to open this up for a discussion. As an instructor, I really like, you know, interchange with students. So I'm going to periodically stop, not just for questions, but for input from you guys. So I'm not sure, Bruno, how you want to do that. If you want to, people maybe have people write something in the chat and then recognize them individually, but we'll work out a little system. So I encourage input on from everybody, and not just questions. If you have any comments on how this might be used in an apologetic context. So here's a short little video. I have two grandsons, and a few weeks ago, I was helping them. You know, summer, you got to keep little kids busy. They're 7 and 9. So my wife and I do a lot of projects with them, and here was one that was a little frustrating until we got it to work. So this is about, I'd say 15 to 20 centimeters tall. It's walking around on their back patio. It took me a good, and my grandsons, a good about 2 hours to put this thing together and to get it to work. Right, here's just, you know, it came in a kit. Here are some of the parts. Had over 70 plastic pieces, some gears, pinions. It was powered by a little electric motor connected to a solar cell. I'm not the greatest with machinery and things like that. I did the best I could and was able to get it constructed and actually to work. I made 2 of them for both grandsons. The first one worked fine. My first trial through it. I think it had about 6 or 7 gears. It has wheels. The second trial was really a trial because I couldn't get it to work, but after a lot of problem solving and a lot of rebuilding, I did get it to work. So I'm sure you've done things like this. Some of you might be involved in engineering all the steps. These were the pieces. You had to cut them out. They're all formed. They had to fit into different slots. There, of course, was a numerical order. You had to go by these steps. Do not deviate from any of the steps. The second time when it didn't work, I thought I had done all the steps and I had to rebuild it several times, but I finally got both of them to work. More instruction pages. There was a main gearbox, the solar panel, and then the dinosaur was really cute. One of my son loves dinosaurs. It actually, you could see it moving and walking. So this really got me thinking, because at the same time I was taking the biochemical design, how machines work, how they're designed with a purpose, how all the parts seem to fit together in a certain order. This whole idea of specified complexity. So this was the kit. It was called Metaphor Teach Tech. It came with a detailed instructional manual. As I said, it was solar powered. And actually, you could actually build 4 different types of robots: a dinosaur, a beetle, a drilling rig, and kind of they call it a quintessential robot. Over 70 different plastic parts. So we're going to kind of come back to this idea and talk about this idea of design. What are the qualities that we can distinguish in man-made human constructs and designs, and how we might be able to distinguish those from natural products. So a few scriptures we all are probably again familiar with. God reveals himself through creation, through his written word, and through the living word of God, Jesus Christ, our Lord and Savior. Romans 1:20, for ever since the world was created, people have seen the earth and sky. Through everything God made, they can clearly see his invisible qualities, his eternal power and divine nature. So they have no excuse for not knowing God. You made all the delicate inward parts of my body and knit me together in my mother's womb. Thank you for making me so wonderfully complex. Your workmanship is marvelous. How well I know it. You watched me as I was being formed in utter seclusion, as I was woven together. Let me move my thing here. Together in the dark of the womb. In the beginning, the Word already existed. The Word was with God, and the Word was God. He existed in the beginning with God. God created everything through him, and nothing was created except through him. The Word gave life to everything that was created, and his life brought light to everyone. The light shines in the darkness, and the darkness can never extinguish it. So as Christians, we main tenet of our belief, our faith, is that God is responsible. He is the creator, designer of the universe, and all life down to the minutest detail, even in the biomolecules and machinery of the cell. Okay, as far as what does the Bible tell us about how God and what God created? And there's a variety of scriptures that especially many of you, I'm sure, know about through reasons to believe in your own studies. But as far as life forms, Genesis talks about vegetation, fish and other ocean life, birds, everything that scurries and swarms in the water. A lot of people think that's talking about and alluding to the Cambrian explosion and all the invertebrates and simple life that filled our oceans early on in creation. other animals, livestock, small animals that scurry along the ground, wild animals, and of course, humanity on day 6. So what about the origin of life? Okay, what does the Bible tell us about that? And it's kind of unclear. This is a verse we talked about quite a bit in the class I just finished. And it's the last part of Genesis 1:2, so it's labeled B. And the Spirit of God was hovering over the surface of the waters. And that comes after, of course, when the Bible talks about the earth was formless and void, and darkness was over the covering over the earth. So this may be an allusion to the fact that God was beginning the process of creating life. Okay, two competing worldviews that we're familiar with, and that is naturalism. Nature is all there is. Natural processes account for both the physical universe as well as life itself. Humanity is only one of billions of life forms. This is a famous statement, I'm sure everybody's heard of it, by Carl Sagan. The cosmos is all that there is or ever will be. Of course, that stands in contrast to biblical creationism, where God created all the universe and all life forms with a purpose. We are unique and special, made in the image of God, the crown of all of God's creation. And of course, Jesus Christ restored humanity's broken relationship with God through his death and resurrection on the cross. Okay, now what do these two differing viewpoints say about life's origin? And I'll stop after this and take a few questions. The idea in naturalism is referred to as chemical evolution or selection, and it's a process that's believed to have taken about 1 billion years. And basically, we start with simple inorganic molecules. Then from those, organic molecules based upon carbon were formed. The building blocks of the 4 kinds of major biomolecules, which are carbohydrates, lipids, proteins, and nucleic acids, are called monomers. And those are things like amino acids, simple sugars, fatty acids. So those were believed to have formed in this chemical soup, which, by the way, probably a lot of you know that there's geochemical evidence that this soup probably didn't even exist. But then monomers, these monomers, in order to form these big giant molecules of which we're made, must be turned into polymers, which are like strings on a bead. So for instance, a protein would think of just a necklace or a bicycle chain. All those links or beads would be amino acids. That's a polymer. Okay, and then these polymers, we'll see in a minute, take different shapes and structures, and those structures give it functions. Okay, but we still don't have anything close to life. And then you have these molecules being encapsulated by membranes with isolated chemical activity occurring inside there. You still need information and metabolism. Okay, that includes energy and energy processes. And after that, that gives rise to simple cells, prokaryotes, bacteria type cells, and eukaryotes. So this is still a mystery. A couple of Fuzz has written some excellent books and articles on the origin of life and the different ideas of how this may have come about. There's lots of research being done, and there has been made some progress made in some of these steps. The leading hypothesis is called the RNA world hypothesis, but all these steps, however and whenever they occurred, were random and undirected. Okay, the contrasting view, RTB's model for life's origin, is called special creation and involves God's direct involvement as the master creator, designer, biochemist. So I'd like us to, if not now, a little bit later, just get some input on that idea of what it means, God's direct involvement. I have an article coming up, and I can also make available to the link to this article. Some of you may have read it. RTB, especially Dr. Rana, emphasizes this idea, which is called hyper-naturalism. It was first put forth by two writers, Dyke and Hendry. And in this, I've got it in red, they basically posit that God employs the laws of nature, including the chemistry, biochemistry, and chemical processes he designed in an extraordinary manner with respect to timing, location, and magnitude to accomplish, in this case, life's origin. Okay, continuing on with this RTB model for life's origin, in essence, and this is something we could also talk about, and maybe you guys have discussed as a chapter. When origin of life researchers perform prebiotic simulation experiments, they operate in a hyper-natural way. They control the experimental setup and laboratory conditions to bring about extraordinary circumstances. However, this is within the confines of the laws of nature and natural processes, thus bringing about key steps in the pathway to the origin of life. Could it be that God functioned like a divine organic chemist when he brought life into existence? In light of this question, it is intriguing, so this is a direct quote from the article, that the Judeo-Christian scriptures describe humans as made in God's image. If that is the case, then when we create or when humans step into the laboratory to run a chemical reaction, are we mimicking, though imperfectly, the Creator? Okay, and one more slide and then I'll stop here for a couple minutes. Origin of life and synthetic biology researchers are thus actually providing us with insights into how God may actually have created life and its amazing biochemistry. So as I mentioned, the building blocks of proteins, these little beads on a chain, are amino acids. Some people take amino acid supplements. Proteins are a major component of all living organisms, but they only come in two forms. There's what's called a left-handed form and a right-handed form. And this is called the problem of homochirality. Amino acids and proteins are left-handed. These are what are called mirror images of molecules. So if you put your hands up, right, there's no way to get the left hand to look exactly like the right hand. However, they are mirror images, and they actually diffract light in different ways. So amino acids and proteins are left-handed, sugars in nucleic acids are right-handed, and that's in living organisms. That's in living organisms. You actually find mixtures of these in nature. But in us, in cells, and all living organisms, they always must have these different forms. So far, there's a lot of research being done on this because, of course, you have to have proteins, right? Remember in our flow chart there, we were talking about polymers, and they all have to be either left-handed or right-handed, depending upon whether they're proteins or nucleic acids. So there are these possible mechanisms that researchers are looking at, but I believe about the highest purity they get is 20 to 30%. And so this is a severe stumbling block to origin of life researchers and also synthetic biologists, at least if they're working from the bottom up. But that doesn't mean there are actually some Christian molecular biologists that feel that in the next 10, 20 years, molecular biologists will actually solve this problem as far as a mechanism by which these molecules can reach large homochiral concentrations. Okay, so we'll just take a little break here before I get into the real meat of my presentation on the watchmaker argument for the ribosome and for the ATP synthase enzyme. Anybody want to make any comments so far on what I covered? Questions or comments? And Bruno, I'm not sure how you would like to do this part.
Anybody that wants to make a comment, just unmute yourself and say whatever you want to say or ask a question.
Hi, Rick. I was just wondering where you took this course.
I was through Reasons to Believe. They offer it once a year, sometimes in the spring and sometimes in the fall. It's a very comprehensive course. I believe it's either 16 or 18 weeks. It's really outstanding. It's through reasons.org.
It's an online course.
Online course, yeah.
I was looking at your timeline there. I was discussing with my buddy Robert just a few days ago, and I was looking at the cell development from 3 to the present of 3.8 billion years. And I said, if we take that 3.8 billion years and we have 38 trillion cells in our body, that adds up to like 10,000 years per cell. Now, if we take the type of cells, we have 200. So now that adds up to 500. So I see a situation where in 500 years, how can they say that we're almost creating some sort of new cell? Like the mathematics doesn't add up to it, and it doesn't add up in what evolutionists or what they're trying to say there, because we would be able to see, if it was that actual short of time period, we would be able to see dramatic changes, would we not?
Well, yeah, I think that the main point, you're on to one of the main points, and that is Dr. Rana discusses this, so does Dr. Ross in their book Origins of Life, and it deals with the time frame and the difficulty in assembly of these biomolecules, from the simplest to the more complex, and some of those steps that I went through, which again is a model. But even at a billion years for all this to happen, it just, and it's amazing to me. Again, I'm not a biochemist, but they say that that's not even enough time, you know, because all this was done according to naturalism. There was no direction. It was all totally random. And there are so many conditions on the earth that would actually favor the disruption and the degradation of these molecules. But but then going on from prokaryotes to eukaryotes, that took another, prokaryotes are like bacteria, which are considered simpler cells. And then we're, of course, like you were alluding to, we're eukaryote cells, much more complicated biochemically and structurally. So that is believed to have taken in the naturalistic model, again, a long period of time. I think it's like 700 or 800 million years to go from prokaryotes to eukaryotes. So time, you're on the right track. Time is a serious concern. Even though, like we say that the Earth has been around for 4.6 billion years, that's really not a lot of time.
Well, I found it very interesting because I can do the basic math on that. I mean, to actually bring it into probabilities factors and stuff like that is mathematics beyond my control. But if there's anybody there that could do that and give a better understanding of what the chances of that happening, I just look at the fact that according to the basic math I did, we should be able to see major changes happening like every 500 years in cells in development, and we're not. We've been around for thousands of years. I mean,
the situation is actually even worse than that, because Hugh and Fuzz, probably, I think they talk about it in the Origins book, that there was a period of time called the late heavy bombardment. So that, part of the problem is that the physicists don't talk to the biologists. So the biologists are not aware that this occurred. And so the Earth was basically habitable until about 3.8 billion years ago, because it was all molten lava. And at that point, it cooled off, and they found evidence for bacteria right at that same point. So there was no billion years of time. There was only maybe a few million at the most. So the time period is a lot less than what was originally believed.
Yeah, a very good point, Bruno. Thanks for bringing that up. That's exactly correct. Okay, is it okay to go on then? Any more comments or questions?
Anyone have any?
I was just wondering where the hope for solving the homochirality problem is.
Well, yeah. All these mechanisms are being explored, heavily researched, and I would say it's somewhere in that last little section. That's why I went back to this slide that deals with this idea of there are ways to produce. If I'm remembering correctly, like 20 to 30%, which isn't a lot, but it's better than, a 20, I'll say it this way, a 20 to 30% excess. So instead of a 50-50, what's called racemic mixture, you might get, 60 or 70%. But you have to have 100%. So they're getting there. And I think, again, RTB, especially Dr. Rana and other molecular biologists are cautious to say that it just can't be done naturally, because it seems like creationists have been wrong in other instances. So I think my perspective is that RTB is very cautious in some of these areas, because we're learning and making so much progress so quickly. It may not happen, but it may. But I think it has to do with this. If they can produce some of these excesses beyond the 50-50 mixture. Okay, so going on, is it okay to go on?
Sure, go ahead.
Okay, so William Paley, back in the 1800s, late 17 to 1800s, proposed this idea, which is called the watchmaker argument for God's existence, based upon the intricate designs we recognize in living organisms, and felt that it's fairly intuitive for most of us to pick up something or look at something. He used a rock and a watch to tell that it's been designed. It's an artifact. It's a human construct, as opposed to a result of purely natural processes. And he was looking at organs and organ systems, kind of the beginning of anatomy and physiology. But now, again, one of the things that just amazed me is we can go at the molecular level. Now, biochemistry is so advanced that we can actually find evidence for design qualities in biomolecules, biomolecular complexes, and cellular machinery. Now, to be fair, there are two criticisms of this argument that if you ever use it, you need to be aware of. And one is most biochemists do recognize design, but they call it an appearance of design. It's designed, but it only appears that it's designed, which is kind of interesting. So we have to, as we have there in red, can we demonstrate, is there a way to truly demonstrate logically, scientifically, empirically, that the design we see is real and purposeful? And then a very early criticism was proposed by Hume that it's a faulty analogy. It's a faulty comparison. But there's an idea that I first heard of in my class, and it's called, let me move this here, the von Neumann self-replicator, which is basically, imagine a computer that could actually manufacture computers just like itself. At this point, it's a theoretical construct, but engineers are actually working towards achieving this idea of this von Neumann self-replicator. And so there is this comparison that we can make between cells and biomolecular systems that replicate themselves and human constructs that eventually may be able to do the same thing. Okay, everybody's probably familiar with the blind watchmaker argument. Richard Dawkins was a big supporter of that. So can science detect intelligent design or intelligent agency? Science and science-related disciplines already do it, such as in forensic science, trying to determine human interference and or causations of death. CT, the search for extraterrestrial intelligence, patterns, information in radio signals. Archaeology, of course, uses logic and methodologies for determining evidence for intelligence, culture, tool use, technology, art, language, symbolism. Archaeologists can look for distinctives to determine even how advanced a culture is. So can we do this at the molecular and biochemical level? Okay, Dr. Rana proposes what's called a three-pillar approach to establishing intelligent design in biochemical systems. One, challenging the mechanisms of natural and chemical and physical processes to produce the construct. Good example of that would what we were just talking about, the homosexuality problem. Analogical reasoning, that's the basis for the watchmaker argument, establishing the case for actual design based upon some type of filter or appropriate protocols, and empirically testing in the laboratory, do some of these biochemical systems actually work as machines? Okay, and within that also the role of intelligent agency. There are a couple new articles on the RTB website that deal with this idea of the hand of God dilemma. In other words, biochemists are tinkering perhaps the same way that God tinkered with biomolecules in creating the first life on our planet. So the whole idea is, can we as Christians use what we're going to talk about now to demonstrate the existence of God? And Hebrews 11:6 says, and without faith it is impossible to please God, because anyone who approaches him must believe that he exists and that he rewards those who earnestly seek them. Last year, for about 2 or 3 months, I had an ongoing online conversation with an atheist. And what I was trying to emphasize, not just biochemically, but from all different aspects of science, was that there are scientific evidences for God's existence. And then to move on from then, if God existed, then what type of God, who is he, leading to the plan of salvation? Okay, so design attributes. These are some of the attributes and things we can recognize from human-based designs. And since we're made in God's image, we would expect these same attributes, these same characteristics to appear if in fact biomolecular systems are designed. Irreducible and specified complexity, fine tuning, complex algorithmic information, the chicken and egg paradox, optimization within appropriate constraints, ingenuity, forethought, and planning, repeated use of the same design or design qualities. Okay, so focusing on now ribosomes. Ribosomes are the protein factory of the cell. All types of cells have ribosomes. This is an interesting quote I found. Life began when ribosomes appeared. So going back to the very first, most primitive life forms, they had ribosomes. Okay, the second complex that I'm going to talk about is ATP synthase, and it is found in the mitochondria, which is the powerhouse of the cell. This is one of the organelles inside of cells. It's also found in inner bacterial membranes, and it's basically an enzyme stuck in a protein. Okay, so ribosomes, again, are the protein factories of the cell. There's what's called the basic flow of information of a cell. And I'm sure you know DNA has the chemical code of life. That code is what's called transcribed into messenger RNA. And then messenger RNA takes the code to the ribosome where proteins are built. And basically proteins make us who we are. They're the most diverse structurally and functionally of all the biomolecules in a cell. Okay, the ribosome comes in two subunits, a larger and a smaller. They're slightly different in prokaryotes and eukaryotes. It's made mainly of ribosomal RNA. Notice the number of proteins, a huge amount of proteins, 2 and 1 in prokaryotes, 3 RNA molecules and 1 RNA. Again, this is in the larger subunit, the smaller unit, and over 80 proteins. So they're very, very complex structures. Okay, this is a cartoon. We're going to watch a video in about a minute or two, and you're going to see that another form of RNA called transfer RNA brings the amino acids to this kind of factory where they're put together. These are abbreviations for amino acids such as alanine, serine, histine, glutamine, glycine. So here these amino acids are coming out of the factory, if you will. Here's the directing information, messenger RNA. The transfer RNAs bring individual amino acids one at a time. They're linked together in this factory, okay? And only about 20 years ago, through crystallography and X-ray diffraction, biochemists are actually able now to get three-dimensional structures, pictures, diagrams, and computer simulations of ribosomes. And this just kind of like blows me away, the complexity of these things. So these strands, these are the RNA molecules. And again, I can't remember if this was a eukaryote one, but this is the upper unit. So there would be 3 RNAs. There would be roughly 50 proteins. These little dots are actually atoms, and they're in globular proteins, which also have a 3-dimensional shape. So remember that little, my little robot, all those plastic pieces that went together? That's the idea, okay? Here's the lower piece right here. Functional ribosomes have to have both. Okay, once a molecule is synthesized, so this would be a chain, this would be rRNA, and it has like ladder structures like DNA, but then it has these loops. These are actually synthesized in a special part of the cell called the nucleolus, inside the nucleus. And then these now bend and twist due to hydrogen bonding and ionic reactions on the functional groups of the molecule. So they twist into this 3-dimensional structure, which is so critical to the function. So this is an RNR. This is one of the rRNA molecules right here, showing the secondary structure, and then the, showing the primary structure, then the secondary and tertiary structures. And this is transfer RNA, the one that carries the amino acids. So here you can see, again, these would be nucleotides, and they bond, there's base pairing here. So they form these ladders and loops, and then this thing begins to twist and turn. And so this would be its tertiary structure. So the structures of these things are highly specific, just like a 3-dimensional piece of plastic or a model. Okay, this is just to show you a little bit, so I wasn't sure of the biochemical background that people have. Proteins and nucleic acids start as chains. They then fold into a helix, or what's called a pleated sheet. Then they curl up on themselves, and then they also actually can bond with multiple polypeptide chains before they actually become functional. How these bond together is through a simple what's called dehydration reaction, in which water is removed. This would be one amino acid. This would be another amino acid. Okay, here's one of my biochemical jokes. Okay, what is a pirate's favorite amino acid? I don't know if you guys have heard of this. Arginine. Glad I heard a little chuckle out there. Binding of nucleic acids. Notice, remember when I had that ladder picture? It had lines across the ladder, like the steps of a ladder. Well, again, a little review of your biochemistry. In nucleotides, there are bases. Adenine always bonds with thymine. Guanine bonds with cytosine, except for in RNA, thymine is replaced by uracil. So these bonds are critical, and as these are in RNA molecules, RNA is a single chain, but remember how it twists and turns, and then let's say an adenine can bond with, this would be a uracil, and a cytosine can bond with guanine, so that forms the ladder, and then some of the loops, these interactions between the bases. Now, one thing we learned in the class that I wasn't aware of is, and this to me is like totally mind-blowing, and that is that ribosome subunits are pre-assembled inside the nucleus, but their assembly is self-assembly. In other words, the subunits made of rRNA, okay, and the proteins, and then there are other proteins that assist in the process. They're all produced either in ribosomes outside the nucleus or in this area that I alluded to before called the nucleolus, and they self-assemble. So the genes have to function in order. These molecules have to be in the right concentrations. They have to meet each other, and this to me is totally amazing. This concept of self-assembly. Dr. Rana shared with us recent research in which there are some engineers who are actually getting close to manufacturing cell phones in a self-assembly line, in which all the parts of the cell phone are actually self-assembled. So that's what happens to ribosomes. They're self-assembled. Okay, this is a video that one of the videos I'm going to show now. Hopefully it'll work. Give you a good overview of the ribosome.
To produce a functional protein, the genetic information found in the form of an mRNA, which is composed of nucleotide building blocks, must be translated into the language of proteins, which are composed of amino acids. This translation process is orchestrated by a large macromolecular ribonucleoprotein machine in the cell, known as the ribosome. It uses a key adapter species known as a transfer RNA or tRNA. tRNAs are the molecules that actually translate the nucleotide sequence into an amino acid sequence. tRNAs are bifunctional in nature, with 3 nucleotides poised at one end of the molecule to read the mRNA template in the
Okay, so that on the bottom, that is the mRNA, the purple one, and this is the tRNA.
anti-parallel fashion. This is referred to as the anticodon end of the tRNA. At the other end of the molecule is the acceptor end, where an activated amino acid is attached. This amino acid corresponds specifically to the identity of the codon being read. Amino-acyl tRNA synthetase enzymes are responsible for catalyzing the loading of the amino acid onto the appropriate tRNA.
Okay, so to get that amino acid onto the correct transfer RNA, there is a special enzyme. There's actually 20 of these. There are 20 main amino acids that make up our proteins. And it's that order of those amino acids that determines the various levels of structure. But these specific enzymes check and proofread and make sure that the correct amino acid, shown right here in green, is actually connected to the correct tRNA, and on the bottom of the tRNA is the anticodon that bonds to the codon.
The fidelity of this step is extremely high, which is critical to the overall fidelity of protein synthesis. In its most basic form, during translation, tRNAs walk along an mRNA in the 5' to 3' direction. As they move, the tRNAs sequentially recognize or decode consecutive 3 nucleotide codons that correspond to a given amino acid. This highly orchestrated march along the mRNA template takes place in the heart of the ribosome, facilitated at each step by portions of this machine, as well as by extracellular factors that participate in each step during the translational cycle.
Okay, this is really important right here. I have a couple slides, but I think I'm going to save some time, skip some of the slides. You'll notice these blue blobs right here. These tRNAs can't really link up and find the ribosome by themselves. They have to be transported by another protein called EFTU.
The ribosome has binding sites for 3 different tRNAs. One tRNA is always bound in the centralmost position in the ribosome and carries the growing polypeptide chain. We refer to this tRNA as the peptide tRNA. It's bound in the P site of the ribosome. The incoming tRNA with the new amino acid to be incorporated binds adjacent to the P site in a site referred to as the A site for aminoacyl tRNA. There is a third binding site, the E or exit site, where the tRNA binds on its way out of the ribosome, following the incorporation of its amino acid into the polypeptide chain. As in most processes, there is a beginning, a middle, and an end. The initiation phase of translation consists of the small subunit of the ribosome working in concert with various initiation factors to identify the starting AUG methionine codon that is positioned in the P site of the ribosome. Next, the initiated tRNA engages the AUG codon and the large subunit of the ribosome joins in a catalyzed reaction. Because of the substantial differences in the mRNA structure between bacteria and eukaryotes, this process is quite different in these two systems.
Okay, that also is pretty quick. That process called initiation also requires other proteins to fit to the small unit, and those are called initiation factors. There's 3 of them.
In bacteria, a polypurine motif in the mRNA, known as the Shine-Dalgarno motif, is found 6 to 8 nucleotides upstream of the initiator AUG. This sequence directly pairs with a polypyrimidine motif in the small ribosome subunit rRNA to position the AUG in the P site.
The reason I stopped here is getting back to this idea of time and the origin of life. How prokaryotes, initiation in prokaryotes is quite a bit different than initiation in eukaryotes. And it deals with this unique sequence that must be on all messenger RNAs. Only in prokaryotes that's totally absent in eukaryotes. So again, how did this transition occur? Molecular biologists may at some time figure out how this mechanism did occur, but it may have taken some type of direct intervention by God, the creator designer, for this huge difference.
Where the initiator tRNA binds to it. In eukaryotes, the ribosome engages the 5' capped end of the mRNA and, in association with a collection of initiation factors, scans along the mRNA until it encounters the 5' most AUG codon. Because the initiator tRNA is part of this scanning complex, it plays a key role in finding the AUG, positioning the ribosome complex where it belongs. After initiation is complete, the ribosome is ready for iterated rounds of amino acid addition. This phase, referred to as elongation, can be broken down into 3 basic steps. In the first step, the ribosome must select the aminoacyl tRNA that corresponds to the codon poised in the A site of the ribosome. This step is facilitated by the GTPase elongation factor EFTU in bacteria, or EEF1A in eukaryotes. Simply put, the process of tRNA selection consists of two phases. In the initial selection phase, an aminoacyl tRNA in complex with EFTU can be rejected or accepted by the ribosome. Once the complex has been accepted, GTP is hydrolyzed and EFTU leaves the ribosome. A near cognate tRNA can still be rejected in a second phase, which is referred to as proofreading. In both steps, thermodynamic and kinetic mechanisms are used by the ribosome to aid in discrimination, such that the system chooses the extremely high fidelity, the cognate tRNA from the 30 or 40 tRNAs in the cell. Once the tRNA selection step has taken place, peptide bond formation is catalyzed in the large subunit. In this step, the growing polypeptide chain is transferred from the peptidyl tRNA to the nucleophilic amine of the A site tRNA amino acid. This transfer extends the polypeptide chain by one amino acid. At this stage, the extended peptide is attached to the tRNA in the A site. This whole mRNA-tRNA complex must now be repositioned in the ribosome to open up the A site for the next incoming aminoacyl tRNA. This translocation step is catalyzed by another translational GTPase, EFG in bacteria and EEF2 in eukaryotes. The GTPase couples the chemical energy of GTP hydrolysis to mechanical motion.
So again, that's a pretty fascinating step to me. This whole idea of now we need another large protein that actually is able to like almost work like a little lever or ratchet to push these over into and help orient them in the right area so that this one will then leave, open up the A site so the next one can come in.
These core events of elongation, tRNA selection, peptide bond formation, and translocation happen iteratively until the ribosome reaches a codon in the A site that signals stop. Stop codons are recognized by protein factors known as release factors that function in an analogous manner to tRNA. One end of the release factor recognizes stop codons positioned in the A site with high specificity, and the other end facilitates chemistry in the active site of a large subunit. In bacteria, there are two release factors, RF1 and RF2, that recognize the three stop codons, UAA, UGA, and UAG, with overlapping specificity. In eukaryotes, there is a single factor, ERF1, that recognizes all three stop codons. The reaction that is catalyzed is a hydrolytic one, wherein the growing polypeptide is released from the peptidyl tRNA, resulting in
So there goes the protein
synthesis. The final step in protein synthesis is referred to as recycling. This step promotes the dissociation of the large and small subunits of the ribosome and the release of the remaining mRNA and tRNA components. At this point, the small subunit.
Okay, I'm going to continue here. Okay, one of the things we were asked to do in the class that I took was to look at some new research, some relevant research to this whole idea of biochemical design. And I found this article in a journal called Science Advances, in which these researchers conducted a 7-year project to basically duplicate this concept of the self-biogenesis of ribosomes. And they actually were able to demonstrate it in the laboratory. It took them 7 years to figure it all out. They actually isolated the genes. This was with a bacterial ribosome. And they put these genes on a certain type of like computer chip in a solution. The ribosome itself has 20 different proteins. So they needed 20 genes for these proteins, as well as genes for the RNA molecules. There are 7, they needed to find 7 more genes that actually assist the process of assembly. These are called chaperone genes. And they had to do it by trial and error. Remember when I was talking about self-assembly? All these parts have to be just manufactured in a certain order so that they can assemble in a specific order in a specific position. And that was what took so long for them to figure out the order, and they eventually did. So this is the whole idea of this concept of the hand of God dilemma. But they duplicated this process in the lab. Okay, I'm just going to show this video, which I think this is an animation that I think will help you appreciate just the whole process and how quickly and some of the conformational changes that the molecules undergo. Oops, sorry. It's not narrated. So we've got there's our messenger RNA. There's the small unit on top, the large unit on the bottom. This is the initiation. That's the first tRNA and the 3 initiation factors. So now we got to start building the protein. Okay, that's called elongation. So now another tRNA comes in, again, transported by this helper EFTU. It's actually tested for a fit. Actually, the smaller subunit communicates stereoisometry through stereoisomerism with the top unit. Okay, then there's the next protein EFGU that ratchets and pushes them over. The peptide bond forms between these 2 transfer RNAs, and the A site is now open. Okay, the third one exits. I love this little motion right here. Gives you a good perspective of this machine. And again, out comes the protein. That's the protein at the top. I'm going to skip some of these slides right here. Oh, just hit a few highlights and then finish up this portion of the talk. There are these specific enzymes that I mentioned that ensure that the correct amino acid is linked to the correct tRNA with the correct anticodon down at the bottom. And what was really interesting to me is this idea of this 3-dimensional shape. The catalytic site is where the amino acid is added, but also there's an editing site, meaning there is a portion of the protein, this is a protein with a quaternary structure, that actually checks the fit of the amino acid. So the catalytic site and the editing site and the where the anticodon is all must line up. And if there isn't a fit, then that's the wrong amino acid and it's kicked back out. So pretty amazing. And then I also mentioned the fact that the 2 subunits communicate through interactions, molecular structural interactions, to ensure that the anticodon and the codons, or the anticodon and the amino acids are correct. So there's multiple checks on this mechanism. Ribosomes are extremely accurate, and that's because of this whole idea of fine tuning and checking to make sure that everything is running correctly. So to finish up, ribosomes, protein generating machines, clearly demonstrate irreducible and specified complexity. You saw lots of different molecules all being vital components. They all have to act in a certain order. They all have to fit. We talked about stereochemical interactions, correct bonding mechanisms, the whole idea of fine tuning, that I mentioned that amino acyl synthase, and how it checks the correct amino acid to ensure that there's not a faulty protein production. It's interesting that molecular biologists have discovered that the rate of protein, or error rate for protein production, is about 1 in, let me move this, 10,000. So that's a fairly low error rate. It could be higher, but they've actually done experiments in the laboratory to speed it up. And if it's higher, then the error rate becomes much higher itself. In other words, if they speed up this conveyor belt machine. But if they slow it down, then they don't get the proteins built fast enough to meet the cell demands. So this idea again of fine tuning, this seems to be the perfect rate, both for protein production to get a kind of an average protein of 1,000 amino acids with an error rate of 90%. Okay, the whole idea of we've got multiple layers of information overlapping, and this is a special type of information called algorithmic information, which is an information that actually produces molecular processing. So we've got information in the DNA code, the messenger RNA code, the transfer RNA. Also there's information in the way the protein folds into the correct secondary, tertiary, quaternary structures. So to me, it's almost like information overload. There has to be all kinds of information interacting with other types of information. The whole idea of the chicken and egg paradox, which comes first? That's a big problem in the origin of life. Proteins are needed to manufacture RNA, but RNA is also needed to be manufacturing proteins as well as DNA. We talked about this idea of optimization within appropriate constraints. The speed of protein assembly is just right to maximize production and minimize errors. Ingenuity and forethought in this process of protein, of building the ribosome and protein production. There are over 100 plus proteins that are necessary just for proteins to be built. And then last, the repeated use of the same design or design proteins. Dr. Rana has written some articles, and I believe he's working on a book dealing with this idea of what's called the biochemical anthropic principle, that many of these molecules and their properties seem just so perfectly designed to produce the processes of life. Okay, so we'll stop here for a couple minutes before I finish up with the ATP synthase enzyme, the cellular power generator. Anybody have any questions or comments? I apologize, I went through that so fast. There's a lot of information there.
Hey,
Any
Rick,
comments?
just one quick comment I'd like
Sure.
to add about the error minimization. One of the key things about the genetic code is that the 3-letter codons that are used for coding for the amino acids have a redundancy built in. And by that, I mean that there are a variety of 3-letter codes that can code for the same amino
Right.
acid. And what's not so well known is that might pose a problem, except that those amino acids which are considered neutral in terms of their charge and don't contribute to stereochemical shape as much tend to have a larger number of codons that code for them. And those that have those that are highly positively charged, like histidine and others, those that are highly negatively charged, like I don't know, aspartic acid, and other highly stereochemical amino acids that may not be charged but are very, very distinct in their shape. They can have only one, tend to have only one code for them. And so this allows for a tremendous amount of variance in the amino acids that essentially don't matter, because you can put in, an alanine or a guanine and you're still going to get the same function and structure. So that's a very interesting property, not just of the coding of the information, but of the actual chemistry of the molecules themselves.
Yeah, excellent point. Thank you. Very good.
Rick, I have what seems like an obvious question, but what exactly is meant by self-assembly?
What is meant by self-assembly? No, I guess the best way I can explain it is if you have the right genes functioning in the right sequence with all, in the case of the ribosome, all the proteins needed, then when those components come together, the ribosome self-assembles without any exterior or extrinsic direction needed. So I'm just thinking of my little robot, that I started off that little video with. If you had all those parts, if you just had all those parts and you laid them out and in some type of container or whatever, they would actually self-assemble.
Was that driven by their shapes?
It's determined by, well, it gets back to that article that I referenced. It's determined, yeah, the shapes fit together, but also the order is critical. The order that these parts meet each other, like when molecules bind, they have to contact each other. So so the right molecules have to be there at the right time and the right space. And for, like at the end, one of my points was basically for a ribosome to be built, you need 100 proteins, you need all those RNA molecules, but they actually self-assemble. Part of it actually happens inside the nucleolus, in the nucleus, and then the two subunits get transported out of the nucleus, and that's where they assemble in the cytoplasm.
Rick, maybe it would help if you sent me a link to that article, which can then be emailed out to everyone later on.
Sure, sure. Be happy to.
Rick, do you mind if I ask a question about, like I'm a computer engineer and network engineer by background. So when you translate this whole analogy to basically modern computer systems, modern networking systems through fiber optics, whatever, what's the analogy of that? Then take us to like what it's like in terms of human development, from a man and woman coming together in conception to produce a baby in literally like 9 months. What kind of miracle is happening there, and the rate of which communications and cellular development is happening? It's astronomical, is it not? Like a star formation.
Yeah, I couldn't agree with you more.
But can you help us understand what is actually happening in terms of the speed of which these little like molecular structures are operating, and the speed of which things are developing through cell formation? I have no concept of how fast it is actually. Do we know?
Yeah, the rates, I'm trying to remember, and I might have to look it up, but I think the ribosome, well, I don't even want to say to give an inaccurate rate, but I know it is mind-boggling. I'm not sure how many proteins, you know, shoot out of that per minute or per hour. I know it is extremely fast. So
I can tell you that in prokaryotic systems, the speed, Rick, is about 3,000 amino acids per second.
thank you. Yeah,
It's really
pretty
fast.
fast.
Yeah. And just another comment, just to clarify, I think once these subunits are made, it's just hydrostatic reactions that brings the various protein subunits together. They will naturally do so because of their hydrostatic charges. What's amazing is getting that subunit built in the first place. But if you throw them together in the right order, as Rick said, they will self-assemble. You don't need an enzyme to make it happen.
Well, one of the amazing things to me that I didn't realize is that some of those proteins that are needed for that assembly inside the middle of the nucleus, those are actually, get this, those are actually made from ribosomes outside the nucleus in the cytoplasm. And those are transported through the nuclear pores into the nucleoplasm. So that to me was pretty amazing as well. I mean, this whole assembly process and the feedback.
Rick, someone asked a question in the chat. Are there any diseases that result from a high error rate in protein assembly that you know
of? Let's see.
Or Don, if you know the answer.
Yeah, I'm sure there are. You know, many diseases are associated with proteins. Whether or not it's probably overproduction of proteins, certainly underproduction, but I would think also overproduction. But that's a good question. I don't know the answer.
Diseases typically are a result of incorrect coding right at the DNA level, not necessarily at the protein level. Examples are, cystic fibrosis is just a single nucleotide shift on the DNA molecule on a specific chromosome causes cystic fibrosis. Lesch-Nyhan syndrome, which is a very bizarre and very rare disease, thankfully, is also caused by a single frameshift mutation. But errors in protein production are definitely, there's quite a few that can give rise to that, but they tend not to be as debilitating as those that are of a genetic origin.
Thanks for answering that.
I would love to ask a question. I don't know if you want to park this one, but forgive me for this question. I'm sorry, but in obvious context of what's happening with the the whole vaccines and the level of which that they want to manipulate the RNA and DNA and the messenger RNA. Forgive me for this. We don't need to answer it if we don't want to, but if we're starting to manipulate things at a molecular kind of that level where we're talking about, aren't we like, is it the safety of this and the effectiveness of this and the long term of this, is it not kind of really dangerous ground? And what's our safeguards around this? And I don't know, we don't need to answer this, but it's something that I'm really concerned about myself. We can talk about it offline, but sorry about that one.
I've
Oh,
got a
that's it.
long paper I wrote on that, Robert, that I can send to you. Rick, go ahead and answer too.
No. Good, great. I think Dr. Rana has addressed that question in some of his articles and even some of his talks at RTB that you might be able to access.
That's really helpful. And thank you for that so much, Don, and also for Rick. And how would we access that? Is that going to be sent out through Bruno, or is that just obviously private information, Don, that's only for certain eyes, or how would we go about that? There's obviously some privacy and confidentiality around that, likely.
I'll send my paper to Bruno and he can forward it to those on the call.
Thank you so much, sir. That's beautiful. Thank you.
I just wanted to make a comment about synonymous codes and the redundancy, but I've read an article at RTB some time ago where they were talking about the synonymous codes and saying that there are some differences, even though you're producing the same amino acid from different codons, the rate at which you produce them can vary depending on which codon is producing a certain amino acid. And when that happens, is what takes place is that the actual shape of the polypeptide produced can be different depending on which specific codon generated that amino acid. Even though it's the same amino acid, the polypeptide will take a slightly different shape based on that. And because it has a different shape, it will actually have a different function. So I'm not sure if anybody else is familiar with this article, but it's something that balances off with the redundancy, because I know the redundancy of the code is also a very has good apologetic value, right? But there's also a matter of the transfer RNAs. I think there's actually 40 transfer RNAs. You can actually have different transfer RNAs for the same amino acid, because again, you have multiple codes for some of the amino acids. So there's a certain function that requires more than just 20 codons. It's not just, we could have gotten away with 20 codons. We actually need more than 20 codons, even though initially we only need 20 amino acids. Does that make sense?
Yeah, I think that's a good point. Yep. I remember. I was just going to allude to. There's a whole group of biologists that are investigating this whole idea of coding, in biology, the levels of coding at the molecular level and the different types of codes. So I think that fits into exactly what you're saying, and how those codes can be so specific and make, a significant difference.
All these videos and all the good comments that you're making. We're talking about simplifications. Everything that we're looking at is actually very simplified compared to the actuality of
Yeah,
how complex, how truly amazing the systems
right.
are. But you know, we're just starting to get at the very bottom level of a very high mountain that we have to climb of knowledge, right?
Very true. We're scratching the surface. Can I take just a few more minutes before we kind of finish up here?
Rich, I think you might want to continue with the last part of your presentation now.
Okay, great. Okay, so this is a computer animated photo of ATP synthase enzyme. And that's my second molecular machine I'm going to be talking about. This is a lot shorter. It is part of a bigger system. It's linked to what's called the electron transport system in the mitochondrial membrane. So these are proteins. You don't have to worry. I'm not going to talk about those proteins in this complex. But basically, it's part of cellular respiration. We're going to focus on this last protein right here, which is responsible for the production, most of the production of adenosine triphosphate, which is the energy currency of the cell. Okay, it consists of actually two types of motors or machines in one. So there's an upper part that kind of resembles a mushroom and a lower part that's actually been likened to a water wheel. So these are all proteins. This is the mitochondrial membrane. Part of it, which is referred to as the FO portion, is embedded in the membrane. The other part extends into the matrix, which is the middle of the mitochondria. And this is the F1 portion. So it says the FO portion consists of proteins. They're simply called A, and here's B, and 9 to 14 copies of protein C, which you're going to see in a minute, acts kind of like a water wheel. The F1 portion always reminds me of kind of like a mushroom stalk. It has alternating alpha and beta subunits and a central gamma protein that actually goes up inside here and down into the water wheel. Okay, I think I'm going to skip most of that. Other than interesting, it generates over, talking kind of about like speed or what these things do, 1,000 kilograms of ATP daily in humans. Okay, the top portion, these are again are all proteins. This is where the catalysis of ATP is made or is produced. ATP is made from ADP and adenosine diphosphate and phosphate, which you're going to see in a minute how they fit into these dimers. Okay, inside here is a shaft. It's likened to a cam shaft that spins and opens and closes these in a three-part mechanism. This holds everything in place. It actually does change its shape, but it's necessary to stabilize the whole mechanism. It's called a stator. And this functions like a water wheel or turbine. And I mentioned this extension, the camshaft. This is a protein reservoir in which hydrogen ions, essentially proteins, enter the bottom side, exit the top side, and they actually cause this to rotate. So this is a pretty short video, but it'll give you a good idea of how this works.
Called ATP or adenosine triphosphate. ATP stores chemical energy in the form of a high-energy phosphate bond. As the name suggests, it consists of an adenosine ring with a chain of 3 phosphate groups attached. The last of these phosphates can be split off to release the energy within the bond, resulting in adenosine diphosphate and free inorganic phosphate. This energy is needed to keep cells alive and is used from processes from muscle contraction to producing the thoughts your brain is having right now. The hydrolysis of ATP can be reversed by the addition of inorganic phosphate to ADP. However, this of course requires energy and is performed by a molecular power generator called ATP synthase. Here we see an atomic model of F1-FO ATP synthase sitting in a membrane. This molecular motor is found in the mitochondria of eukaryotic cells and is responsible for making most of the ATP. Mitochondria, the powerhouses of the cells, turn over around 60 kilograms of ATP every day in an adult person. A long chain of biochemical processes convert the energy stored in food into a proton gradient across the inner mitochondrial membrane. These protons drive a rotation of the turbine within ATP synthase, resulting in the synthesis of ATP. Like all generators, ATP synthase consists of 2 separate motors coupled together. Within the membrane, we have the FO motor, named after the binding of antibiotic oligomycin, and at the top we have the F1 motor from factor 1. The FO motor is a proton-powered motor. It is thought that protons flow through a channel open to just the intermembrane space, where they bind to a ring of protein subunits, rotate 360 degrees, and exit through another channel exposed only to the matrix. The net flow of protons driven by the proton motor force provides the energy for the generation of rotation. The torque generated in the FO motor is transferred to the F1 motor by a central stalk or shaft. The F1 motor is responsible for generating the ATP by addition of phosphate to ADP. The top of the central stalk acts similarly to a camshaft, so that as it rotates within the F1 motor, it causes conformational changes of the catalytic subunits. The catalytic unit is made of a dimer of subunits, and there are 3 of these arranged in a ring. Catalysis occurs at the interface between the dimers. If we concentrate on one dimer, we can observe 3 distinct states. First, ADP and phosphate bind to the catalytic site. The central shaft then rotates 120 degrees to rearrange the molecules. Next, the enzyme undergoes a further 120-degree rotation, and the ADP and phosphate are fused together to create ATP. The enzyme then rotates again to return to the starting position, where ADP is released and ADP and phosphate can be bound for the next cycle of catalysis. One key aspect of this relationship is that catalytic subunits must remain stationary with respect to a rotating central shaft. This task is performed by a scaffold on the outside of the complex, referred to as the peripheral stalk. Recent work has shown that what was once thought of as a rigid scaffold is actually dynamic and is able to accommodate the changes necessary for most efficient function. Looking at this machine in its natural environment by electron chromatography of intact mitochondria has shown that ATP synthase dimerizes to shape the mitochondrial inner membrane into their signature cristae shape. This turbocharges ATP synthesis by focusing the proton gradient near ATP synthase.
Right, hopefully that gives you a good perspective of the workings and how this functions. So to finish up the structure and function of this, as we went over, it's made of 2 motors linked together. Let's see, pretty much the video covered all of this. Chemical energy is converted into mechanical kinetic energy, then rotational energy, then back to chemical energy. Remember, one of the things I mentioned early in the talk is how can you actually demonstrate that these things are true machines? And a recent study was done in which some biochemists actually attached fluorescent actin units, which is a protein, another protein, were attached to the F1 units, ATP added, and this thing whirled around like it reminded me of like propellers. like propeller blades whirling around. So it definitely demonstrated the machine-like properties of the ATP synthase. Here's one of my last jokes about enzymes and substrates. You are the substrate to my enzyme, and nothing can ever denature us. Okay, so ATP synthase clearly demonstrates some of those properties we were talking about: irreducible and specified complexity, all those proteins functioning together, the two motors linked together, demonstrating planning, forethought, ingenuity, the chicken and egg paradox. This ATP synthase is intimately linked to the electrons transport system, proteins, coenzymes that carry hydrogen proteins. They all have to fit together at one time. Optimization, feedback, and fine tuning. ATP production is continually monitored to meet cellular organ and organismal requirements. Okay, so hopefully some of the ideas we discussed here will help all of us build bridges to skeptics and those who have not considered the truth claims of the Bible. All this is from God, who reconciled us to himself through Christ and gave us the ministry of reconciliation. As you know, we are considered Christ's ambassadors. So my prayer is that all of us will continue in reaching out the best we can to those who have not found Jesus Christ as their Lord and Savior. The ribosome and its associated biomolecules, machinery, and ATP synthase complexes clearly demonstrate properties of intelligent design, forethought, and planning. Such properties point to the master biochemist, designer, and creator, and his active involvement in producing these molecular machines and in life and humanity's origins. The identity of the master designer best fits the description of the God of the Bible. All designers have a plan and a purpose for their creations. This plan is revealed in his word, both his written word and in his Son, Jesus Christ. May our lives, actions, and words share this good news with those who don't know him, for we are Christ's ambassadors. And then just to throw it open here for the last couple minutes, I had a couple discussion topics, if anybody would like to comment, no certain order. What do you guys think about this revitalized watchmaker argument, especially using biochemical systems? Can it be effective? Yes or no? Has anybody used it? With whom in particular? Any cautions in using it? And then I'm also interested in comments you might give, because I tried to get people in our class to respond to this whole idea of the RTB model for life's origin. They talk about this term hypernaturalism. So does it go far enough in establishing God's role in life's origin? I don't know if any of you, about a week and a half ago, there was a live RTB presentation in which Jeff Zwernick interviewed Dr. Fuzz Rana. And one of the questions was directed to this. He goes, Fuzz, what do you think personally you would see or witness if you went back to the origin of life? In other words, what would you see happening to all these, biomolecules? What would God be, how would it be acting? And then this whole idea of researchers are making progress, as I mentioned, in origin of life and synthetic biology, but in doing so, are they not giving powerful evidence for the hand of God. This is referred to the hand of God dilemma. And then last, do biochemical systems point towards a biochemical anthropic principle, much in the way bodies in the universe and forces in the universe do? In other words, are some of these biomolecules, do they seem actually exquisitely designed for life and life properties? So would anybody like to comment on any of those 4 discussion topics before we close?
Well, it seems to me the biochemical anthropic principle falls under the general anthropic principle, and that's a powerful apologetic to it. Like, why should there be such a thing?
Back in the 19th century, I think it was, when Paley wrote his book, he said that, life is like a watch, a machine. But it wasn't until they actually got into the cell and were able to see these machines that you could actually make a valid analogy. So I think right now it's more powerful because there actually are machines. There's, rotating motors. There's information. The ribosome takes information from in one form and converts it to another form. So it's like basically in a computer where you copy the data from your disk to your RAM, or from the disk to a CD, or whatever, wherever. You're copying the information from one media to a different media.
Right. Has anybody actually approached skeptics and or non-believers with this argument? So has anybody tried it out? Can anybody give us any feedback on this approach? Effective? Were people somewhat receptive to it? Did they relate to it? Did they make a connection?
I once was engaged with an agnostic person, I guess is how he put it. He's actually from the Sikh faith. We got into a wide range of questions, and that was how the whole thing was. Just ask questions and asked him basically to explain these anomalies.
Mm-hmm.
At the end of it, he told me I was good at what I do, whatever that means. So it can be effective if it's done with the right approach.
Great.
But basically, you're aiming to put questions in their mind that drives them to seek out the answers.
I think that's a really, really good way to put it. Dr. Rana mentioned almost verbatim the way you said that. And that idea of asking questions, people it takes time to think through some of these things. The whole idea of we have this worldview, and it's different, to go from a naturalistic worldview to a biblical worldview. It's a big leap, and they do have to really think through it. And again, I think it's like I mentioned, and Dr. Rana mentioned too, just the role of the Holy Spirit in helping them understand, this argument or this concept of God as the creator designer.
Hugh Ross once said that it takes a long time, also because people want to have good arguments to give their friends when their friends ask them, why are you believing this?
Oh, very good. Yeah, certainly.
Do you mind if I ask a question then to the gentleman that Rich here, and also Don and others, Bruno, that if you scale what we've talked about to the point of which plants have seeds that produce other beautiful plants, animals produce other beautiful animals just by reproduction, human beings reproduce other human beings. The reproduction process of all this is miracles upon miracles. Does this not add even more scalar wonder to this whole thing? There's a real wonder to this, is there not? How do you gentlemen see this? I talk to your doctoral and masteral people in this argumentatively in a good way.
Well, yeah, having when an organism has a child, it's just amplifying this whole process because you're creating a whole new individual. Whereas, the ribosome only takes a copy of one RNA and creates a protein out of it. But when you actually create another living being, you're taking the whole DNA, creating a whole new being that's similar to the parents, or has combinations of both genes from both parents. That's like a million times more complex.
One small point, but it seems to me that this question 2 talks about RTB model for life's origin. It seems to me the naturalists really don't have a model for life's origin. They're largely stumped.
Well, I would respond to that in this way. They have a model, but the model, the best way I would describe it is the model is in crisis. In other words, what they're finding in the lab is not necessarily aligning with their model. And the whole idea of, now, there's the RNA hypothesis, RNA first, but then there's an RNA hybrid protein model that's being put forth. The whole metabolism or information, it's changing. We're learning so much what's kind of possible and what isn't. But they do have a model, but it's fluctuating. It's in flux, and somewhat almost to me, it's analogous to the model of humanity's origin. It's in flux. It changes with new discoveries.
Well, unfortunately, in the case of humanity's origin, I don't see that that's knocked evolution off the pedestal. But origin of life, evolution doesn't even come into that.
How about the question of like when you start scaling these things to like plant life, animal life, reptiles, insects, human beings, birds, they all have different molecular and cellular structures, do they not? They're not all similar to humans. So the fact that we are created in the creator's image and likeness, is there anything that's more divine, the imago Dei, to what we're talking about than the other creatures, perhaps?
You were asking about if anybody's presented this argument to other people, and I'm just recalling that I did. And I ran into the argument of, I forget what the word is to describe the argument, but it's the repurposing of complex organisms that were something else along their evolutionary trail before they became the complex, purposeful organism that we're analyzing and looking at now. So I remember when I took the course in biology, and Fuzz discouraged using irreducible complexity because of that. And I don't agree. I think irreducible complexity is a great argument. And I found that the repurposing, if you can remind me of the word that is used to describe that argument, is a weak argument, I think. And it was used in the flagellum, that there was something similar to the flagellum that was incomplete, and that it had some other purpose before it became the flagellum.
Right. Yeah, I do know what you're talking about, and I can't right now. Trying to remember the term.
I think the term is co-option.
Yeah, very good. Thank you, Hilmar.
And I know that a lot of people like irreducible complexity, and I do too. But I think the point that Fuzz tries to make is that it's effective for people that are already that are already Christians or already sympathetic to the idea. But if it's not that effective for scientifically minded individuals that are agnostic, and so you can use it certainly, but you have to use it in combination with other factors. So that's why he talks about the ID pattern. So irreducible complexity is one of the items in the ID pattern, in detecting the ID pattern. So if you can show that there's irreducible complexity and that there's a chicken and egg dilemma, etc., if you can show multiple pattern characteristics that are indicative of design, it's much more effective than if you just use irreducible complexity. So it's not that he's against irreducible complexity itself. He thinks that in isolation, it has this weakness that can be exploited, as you mentioned, co-option, and all these atheists has to show is that he has a potential solution that that would eliminate this problem for him of irreducible complexity. If he can prove that there's some way that something could be co-opted, he's already won, even though he hasn't actually shown that it happened, but the potential is there, right? So if you use irreducible complexity in combination with other ID pattern characteristics, then you'll have a better chance of convincing somebody that's not already sympathetic to the ID view. So if that makes sense.
I have a question
Well,
here
a couple of
for
other
the
places to go
scientific.
is the information problem, and what was the other one? Why is the natural state of humanity to be religious?
Can you rephrase that again? Sorry, I didn't quite get it.
Well, as I understand it, most people are religious in some sense.
Right.
Why should that be?
Well, my answer to that, and anybody else certainly can pipe in, is the fact that we are, that whole concept of we are made in God's image. We have a spiritual nature, that we have a unique relationship with God, the creator, that animals and plants and microorganisms, invertebrates don't have. And so even though we're fallen, there's still that yearning of that incompleteness, that without God, something is missing. And so that's a unique part of us being made in the image of God, as well as many other properties. Our ability for like symbolism, language, art. We were talking about beauty and wonder. We have lots of unique qualities that many naturalists would argue that we're really not that different. That whole idea of human exceptionalism, but definitely we have that spiritual component that animals and plants don't have.
Well, that's what I would point to as this line of inquiry.
But yeah, it's a good question to raise and see how skeptics or non-believers would answer that. Where does that come from?
I had a
And
question.
the other
for-
powerful argument is the information problem. We marvel at DNA, the molecule, but what about the information it carries? Where does that come from?
Exactly. Very good.
And Brother Ian, just not only the information, but the signaling of information. In computer systems and electronic systems, signaling is very complicated. And the way things are, instruction codes, like the instructions of the information, right? To do what with it, to build what? That's the complicated part of it, the signaling. We haven't even talked really about that in much detail.
Yeah, Dr. Rana really emphasizes that and encourages, if we're going to talk about information, to do exactly what you just alluded to. And it's the quality and level and type of information. And my understanding is they like to refer to it as, you know, algorithmic rather than simple, looking for the words, simple semantic or syntactic information. It's active processing, like you were saying, information, a higher level of information.
That brought me to a question about in this world that we live in with technology and that. And I see this, and I'm not a scientist, so I can't say it from your perspective, but I can see it. I can see it from other sides, like the business perspective, is that we're heading toward this program society where we're given all these processes and steps and everything, and we're starting to follow them, that we're losing our foundation of what the actual truth is based on. And my concern is with the science is that because it's fed into this system, that they're going to dictate what it is, because the guys that control these algorithms and everything else, I'm already, you know, we're already starting to see that, right? Play out in the world.
And just to further the question is, like, algorithmic mathematics is some of the most complicated mathematics we know of. Ramanujan, who is an Indian mathematician, like equivalent to like Einstein, but way beyond his time in terms of abstract mathematics. How did we even have, like, Brother Don, do we even have the mathematics around how the signaling of these, the DNA and RNA structures and messenger RNA actually works and communicates, and they understand each other and signal and build each other? Do we even have the mathematics around these things for it to properly sequentially design it, reproduce it, manufacture it from a synthetic level or a biochemical level at all? Curious about these things. Sorry about the questions, but I'm very curious.
Hilmar, you got to help me out here. I have partly an answer to that question, but I can't remember the term. Remember how Dr. Rana was discussing in one of his lectures, if you look at the structure of the nucleotides, adenine, guanine, cytosine, and thymine, based upon their structure, they've like analyzed it. I know there's like DNA computing, but that's not what I'm talking about. But there's this built-in, and it's a type of computer code. Do you remember what
I think you're looking for the term error correction.
No, there's another term for it, and it's either like, it was either like a binary or a ternary something or other, that when the enzymes can actually proofread to make sure that the bases are in the right order, either in DNA or RNA, they do it through this computer-like processing mechanism, which is,
Yes.
you know,
Odd, or even parity check.
there it is. The parity check. Yeah, thank you. And I thought, whoa, man, you know, that's pretty amazing.
Yeah, so we use it. That's a basic structure of parity checking we used in communications electronics back in the 70s, even before that. We use sylectic redundancy checks now, but it seems very similar to computer technologies. It's almost like, as a computer network engineers, we've taken the human body, we've just manufacture, we just reproduced in terms of systems. But from a level of like at the DNA, we've mapped the human brain. We've mapped the DNA with genome experiments and stuff. So there's a level of this that I guess it's more about bioengineering I'm interested in, and cybernetics, and kind of looking forward. It's another conversation, but evidently men are at a different level with engineering this stuff, and there's purposes and designs that they have in mind that they're going to go with this. Once they understand it, then they can develop it and build it themselves. But yeah, it's interesting where is this all going? You have in Hebrews 1:3, it says that he is purposely moving all things forward. He is propelling all things forward, astronomically, terrestrially, biologically. Everything's moving towards the direction that the master planner, God himself, is doing. So that's also interesting what he's doing too. And he's building a church on top of that, in that process. There's a new birth process. Jesus said, if you would understand the natural things, how shall I tell you of heavenly things? So, right?
Very good.
Seems to me this gets into why Jesus called himself Son of Man. Like you're talking where mankind as a total is going.
I think we're going to have to end this discussion right now. It's about 9:15, and we need to give Rick a break and let him go eat some food. It's probably his dinner time right now. So thank you, Rick, for coming and talking to us and sharing us, explaining to us how these machines work.
Well, it's been my pleasure. Thank you guys very much. Thank you for all your insights and input into the talk. I'll get a couple articles that I alluded to Bruno if you're interested in that. And again, my prayers for all of you and your chapter there in Toronto.
Thank you.
Thanks so much, Rick.
Thank you very much.
So much, everyone.
Thank you. God bless
Thanks,
you
Rick.
all.
Really
Good night,
appreciate it.
everyone.
Thank you. Great presentation.
Thank you.
Night, everyone.
Good night, everyone.